This is an AQA A-level chemistry question, and it's based around the topics of amino acids and DNA. So if you have a look at each question, give it a go, and then once you've tried it, review your answers, pausing as you go. So here's part A, part B and C, and finally part D. So let's go back to part A. Use the information in the data booklet to help you answer these questions. A single strand of DNA is made from many nucleotides linked together. Draw the structure of the nucleotide that contains guanine, showing clearly the bonding between the components. Now, I've actually put up the mark scheme as it exists, as you are hopefully familiar with, or certainly can go to find this information. You need to have the correct phosphate sugar link on the third carbon, and we can see that just here. You need to allow the phosphate attached to 5, and we can see that one there. And we need the correct sugar guanine link on C1. Now, remainder of the molecule then all being correct as well. I think what's important to note here is that there is a huge amount of information that's provided to you in the data booklet. What I would recommend you do is get familiar with that page, because once you are familiar with what the different bits are, all you then need to do is work out where the connections go, and that becomes much more manageable. Moving on to part B, 2 complementary strands of DNA form a double helix in which one strand is attracted to another by interactions between pairs of bases. Complete the base pair diagram showing the interactions. Now, again, we've got here a little bit of information. This is guanine. Now, you have got to remember the complementary base pairs. So we know that guanine goes with cytosine, G and C, A and T. Now, once you've got that and you recognise that it's cytosine, what you've then got to do is remember how many hydrogen bonds there will be and where they are going to be. So again, I've added the answer directly here, and you can see where the 3 links are. This O is relative to this O, this H to this H, and this H to this H. And I've got the cytosine molecule drawn on the other side. If we move on to part C now, how do the interactions in adenine-thymine base pair differ from those identified in part B? Well, there is a really fundamental difference, and it hinges on the number of hydrogen bonds. Guanine and cytosine, we've got 3 hydrogen bonds. But in adenine and thymine, there are only 2 hydrogen bonds between the base pairs. And that takes us to the end of part C. Finally, moving on to part D, urea, we've got the displayed formula provided. Why would urea be effective at separating the complementary strands in DNA? Well, the first thing that we need to look at is that it's got 2 amino groups. It's got these NH2s that I've highlighted. Why is that important? Well, they could substitute for the H bonds in the double helix. That takes us to the end of this question. Thank you for listening.